Now let's take a look at myelination. So we heard earlier, yeah, about formation of myelin sheath. So myelination really is a process of forming the insulation, myelin sheath. So this is the coating around your electrical cords. So if you take one look at one of them now, you should be able to see if the rubber coating of it. Now, so don't forget what cells forms the myelin sheath in the central nervous system. Do you remember that? Right, start with an O. Oligo, right, dendrocytes. Okay, and those that are responsible forming the myelin sheath in peripheral nervous system, right, Schwann cells. Okay. So if we take a look at this diagram picture over here, they took, here is your axon, okay, and this bigger cell outside of it, this is the Schwann cell. The nucleus of the Schwann cell, cytoplasm of it, and then the Schwann cell forms layers and layers of that, of itself surrounding the axon. So here is the myelin axon, but outside of it, here is the myelin sheath. Okay. So how does it form the myelin sheath around it? So if you take a look over here, here is the Schwann cell wrapping itself around the axon part of it. So can you see over here? Here is the plasma membrane of your Schwann cell, and it keeps on wrapping layers and layers around until it forms into many, many layers. So here is your myelin sheath. And that will help with all the information that is going to be passed along the axonal neuron. Okay, so it's very, very important for them to form the myelin sheaths. And over here, you can take a look at this cartoon. Over here, you can see the unmyelinated axon, okay, versus on the right hand side, the myelinated axon. Now, which one can send nerve impulses faster? Right, this one, huh? This is the winner. So if you do have myelination occur along the neuronal axon, information can, especially a nerve impulse, can jump and they process much, much more faster than a myelinated axon. But we do see both of these type of cells, axons, either unmyelinated ones or a myelinated one in our nervous system. So let's take a look now at a region that we call the nodes of Ranvier. So between the Schwann cells, here's another Schwann cell, here's another one, and here is another one. So they leave these open area, which we call the gaps, okay, in between the cells. So those area is your node of Ranvier. And this is where we say, earlier you saw in the cartoon over here, where nerve impulses actually jumps from one place to another one. Yeah. So this is where the node of Ranvier is, and they are the gaps in between the Schwann cells or formation of the myelin sheaths. Now, something terrible sometime might occur later. We'll take a look at demyelination. But first, let's take a look at something that relates still to the formation of myelin sheaths, or the unmyelinated ones. So we have those that we call white matter, and those that we call gray matter in our central nervous system. If we take a look at the white matter of your brain and the spinal cord, you can see them on, for instance, the inner part of it. Okay, these are the white matter. Okay. And these are actually formed from, we say, myelinated axons and neural cells. So as a result, remember, all these myelin sheaths actually are formed from, for instance, lots of layering of the plasma membrane. So there's a lot of fatty materials over here. Okay. As a result, they look more whitish. Okay. More whitish. Okay. More whitish in a way. So As a result, you see the white matter and the central in the brain part of it, and you can still see, for instance, in they are on the outside in your spinal cord regions. Now, the gray matter that we can see on the outer cortex or on the inner side of the spinal cord, these are formed from unmyelinated axons. Okay? Because without all the fatty materials wrapped around it, they look more grayish or so. Now, these myelinations are very, very important. And when you do see, for instance, a loss or destruction of the myelin sheath, we see, for instance, breakage of the nerve impulse. So diseases like multiple sclerosis is an example of where you have an autoimmune disease where the person's own antibodies, okay, or immune cells, and they are actually cells, and they are attacking the myelin sheaths. Okay, are attacking the myelin sheath of your regions over here. So as a result, imagine all your protective coating are gone. Yeah. So what would that affect? Your nerve impulse. Okay. Yeah. So if these sheets are all gone, then nerve impulses are disrupted. Yeah. So imagine your electrical cords that actually connects your cell phone to the outlet. So imagine that cord is broken or it's actually stripped or so, and then that means all the electricity traveling it are going to be leaked out, and this can cause a lot of damages. And in our body, these damages occurs, for instance, affecting, for instance, muscle movements, okay, and sometimes visions as well. A patient with multiple sclerosis might see weaknesses in muscle movement, and sometimes they affect their vision, so they see maybe double vision. So it's not just a one region um, affection of the body, but you see a lot of other places that are affected as well. And it's a progressive disease to continue with the loss and destruction of myelination. So it's very, very important to take good care and be alert on what's happening to your body. Okay. Now, naming differs a little bit in your central nervous system versus peripheral nervous system. So, for instance, right here we call this the cell body, and here is the axon. So, in the central nervous system, the cell body is called the nucleus, but in the peripheral, you will see them calling ganglion for singular or ganglia for plural. For the axons in the central nervous system, we call them tract, but in the peripheral nervous system, we call them nerves, okay, or nerve for singular. Okay. So don't get confused by these names, but they all refer back to either the cell body or the axon part of it. Now, so ultimately, the main meat of this chapter is about electrical signals and how we conduct them. So very similar to the ones we saw before, our muscle fibers, they can start an action potential. We call them, for instance, muscle action potential. Neurons, as also, they are also what we call electrically excitable. Electrically excitable. So what does this mean? This means they can talk to each other by using nerve impulses. So we have two types of these impulse. One is a smaller scale, we call them the graded potential, and another one is your action potential. Oops, not action. Oh, action potential. So we're going to take a look at these two in a few more slides. But first, we're going to take a look at just a general slide of it. So at graded potential, we say usually are much, much confined in, for instance, the dendrites and the cell body of the neuron, and either it gets a dip or it rises up a little. And we'll take a look at what that means. Action potential is either fires or it doesn't fire. So we'll take a look at how it causes this curving to occur. So, but it all comes to the bottom thing is, what is your resting membrane potential? Okay, so here is a cell. Okay, let's orient our cell first. And here is the outside of the cell. Here is your plasma membrane. And here is the inside of the cell. If they were to take a meter and using electrodes to put it all the way into the cell and read it, they will find out that there are differences in charges. So when they read, actually 0 is in the middle, and they will get a minus value, around maybe minus 70 millivolts. So what this means is inside the cell is more negative. Okay, tells you inside a cell is more negatively charged when compared to the outside. Outside is more positively charged. So this is a relative terms. Okay, so for this, they will observe in, for instance, most cells. Okay, they will see a resting potential of maybe minus 70 millivolts. Okay, telling you it's more inside, more negative inside the cell. So the next thing we're going to take a look at is what helps generate this resting membrane potential. Okay, to keep it more negative inside. Okay, so if we take a look at this diagram over here, okay, here is a cell, and most of your cells, and let's orient ourselves again. This is outside. Here's your membrane, the lipid bilayer, and here is the inside of the cell. So what sets up this resting membrane potential? So it's more negative inside and more positive outside. Actually, it's our sodium, potassium, ATPase. And this really is a pump. Okay, so what it does, this actually will take 3 sodium ions and it will transport it from the inside of the cell to the outside of the cell. Okay, so you transport 3 sodium outside. And this happens when you have to use energy because it's outside is already more positive. Okay, so you actually use 1 ATP and then you break it down to ADP diphosphate as well as inorganic phosphate. Yeah. Now at the same time, it will transfer to potassium from the outside and move that into the inside of the cell. Okay, so this is just for 1 sodium potassium ATPase pump. Now imagine you have 10,000 of them. So how many sodiums will you pump out? Right. You gotta do a little bit of calculation. So for 1 ATPase, sodium potassium ATPase, you'll pump 3 potassium out. So if you have 10,000 of them, times 3. So you'll pump out 30,000 sodium ions outside. And how many potassium ions would you pump inside? Right? You'll pump 2 times 10,000 of the pumps. You're going to pump in 20,000 potassium. Okay, so these are all positively charged, but relatively, which side has more positive charge? Right over here. So as a result, they set up a positive environment, so you have more positive charges on the outside, and all of them are because of your sodium ions. Okay, and the potassium that are inside, okay, and as you pump into the cells. Okay, so as a result, the gradient that they set up is tons of sodium outside but very little sodium on the inside. And you'd set up a huge potassium ions concentration inside, but very little potassium concentration outside. Okay, so this sets up a resting membrane potential where it would be a little bit more negative on the inside. Okay, can you guys see that? All right, we'll take a look at these leak ion channels and a little bit about it. So this will relate to later on when these leak channels open, especially during action potentials. Okay, you have all these sodium ions on the outside and you have very little sodium ions on the inside. How would your sodium ion travel? Right, so you gotta imagine this is like a water dam for the sodium ions. Once the water dam for sodium ions open, they go from high to low concentration. They will rush into the cells. Okay. And same thing on the other hand. Over here you see the potassium leak channel. Remember we set up a huge potassium ion concentration inside, very little potassium concentration outside. So when this opens, right, this is going to go from high to low. Okay, so that does stay the same. Okay, so but ultimately, I want you to know what's the action of your sodium potassium ATPase pump, because this is the main thing that helps you set up the membrane potential. Okay, and the action, how many sodium ion does it pump out? How many potassium ion does it pump in? And does it require energy? Okay. All right, so the chemical gradient that we set up, or meaning the concentration that we set up, is an electrical chemical gradient. So if we orient ourselves again, outside versus inside, okay, this was a pump earlier, remember? So it sets up tons of sodium on the outside, very little sodium left. And you have tons of potassium inside, very little potassium on the outside. So there is a chemical part of it because of the ions itself. Okay. Okay. So now, so because of the ions are on either side of it. Now the other part, which you call electrical aspect of it, it's because the charges of the ions. So for instance, sodium ion has a plus charge, yeah, and potassium ion has their plus charges as well. Okay, so together they set up an electrical chemical gradient because of their charges and because of the ions concentration of that. Okay. So this is very, very important because for an action potential to occur, later on they will need this concentration gradient, the differences between them, where it's huge versus very little inside the cell. Now, so let's take a look at some of the ion channels. Earlier we saw one type of it, but there's actually 3 other types, so a total of 4 types of ion channels. So the one we saw earlier are the leak channels, okay, and we saw the sodium as leak channel, and we saw the potassium leak channels. So in all of our cells they are present, and there are actually more potassium leak channels than sodium leak channels. So as a result, through times, when more potassium channels open, they are going to leak out. Okay? So as your potassium goes from high to low concentration, right, it will make your cells more negative inside, yeah, and more positive on the outside. Now we also have these 3 other type of channels. They are more gated. So some responds to a ligand. So respond to binding of a substance, of what we call a ligand. Could be a neurotransmitter, could be a hormone, okay, or ions, or neurotransmitter. Okay. So these are channels that are ion channels, but they will respond to when you have a specific ligand that binds to it. So it has a specific spot for it as well. And then the channel opens up. Now we also have those responded to mechanical stresses, could be sound wave, okay, or based on the light or harder touch or pressure, etc. So they will respond to changes in all of these possible stimuli. So once the stimuli, for instance, stimulus occur like a sound wave, then the channel open up and the ion goes across. Now we also have dose response to changes in voltage. Okay? That really means membrane potential. Okay? So they will respond when there is a switch between the charges across the membrane. Then it will open up or closes. So here is a summary of all these 4 different types. Okay, as you can see, the leak channels, okay, they randomly open and closes. And so for in your instance, your sodium potassium leak channels, they randomly closes and opens. Now, but it's found in all cells. Okay, all of your cells have that. So to try to maintain membrane potential to help with setting the resting membrane potential when needed. Now we also have liganded, ligand gated or mechanical gated or voltage gated ones. So these are mostly present in your neurons. So under more specific regulation. Now, just like our muscle fibers, we said our neurons are electrically excitable. Yeah, so they can they can receive a stimulus and they can send a nerve impulse. So for the graded potential, we say these are for short distance communications only, mainly in the dendrites as well as the cell body of the neuron. Now action potential, this is going to be over longer distance of your body and occurs mainly at the axonal regions. So for the graded ones, okay, you will see that these 2 diagrams shows, let's take a look at the bottom one first. Okay. This shows you have to start with the stimulus. So for instance, with the first stimulus, then you generate a potential change. So graded potential meaning, okay, right over here, you can see your resting potential. Resting membrane potential. So this, don't forget, this is inside the cell is more negative. Outside is more positive, right? So minus 70. So you have to have a stimulus, could be a sound wave, could be anything. And then it sends a stimulus and your cell responded by having a change in the membrane potential. So when it goes up a little bit, this graded membrane potential shows you it has some charges becoming a little bit more positive inside, yeah? As the number goes less negative. And if you have a stronger stimulus, perhaps a stronger sound wave, you send a stronger potential second time. And if you have an even stronger stimulus, the third one, you will see a stronger bulb as well. Okay, so this shows you this is for short distances, but when you have 2, for instance, stimuli that are close enough together, they do, for instance, add up. So this is called a summation. Okay, when you have 2, for instance, the potential added up together before it can rest. Yeah, back to resting membrane potential. Okay, so let's take a look now at a different thing. Now our action potential, this is the main key player that we say occurs over long distances. So action potential occurs, if you're over here, see your resting membrane potential. Okay, resting membrane potential. Okay. So for instance, your cells receive a stimulus, okay, Could be someone tapping you on the shoulder, or for instance, or you actually step onto a nail or so. Yeah. So hopefully it's not the latter case. Yeah. So what happens here is usually it's more negative inside when you start it, more positive on the outside. And don't forget what sets up this membrane potential, right? So what happens now is when the stimulus is strong enough, okay, when this is strong enough, the sodium voltage channels open. Okay, voltage gated channels open. So this sodium channels opens, and remember you set up all the gradients. You have lots of sodium on the outside, very little sodium ions on the inside. So what's going to happen? Right, once this open, this is specifically a channel for sodium ion. They're going to rush, okay, going from high to low concentration. Okay, so as they rush in, all those sodium ions rushes in. Okay, so a lot of positive charges inside. So that's why when the stimulus is strong enough, you see all the sodiums rushing inside and causing the inside of the cell to become more positive. So that's why you see it going from minus inside the cell to positive, like plus 30. Okay, can you guys see that? So that's how, because all your sodium rushing in through the sodium channels. Okay, so later on when they reach the peak, okay, these sodium channels are going to close. Okay, so sodium channels closes. Okay. And this time, your potassium channels, this one over here, the voltage gated potassium channels are going to open. So remember, we have tons of potassium inside, okay, because you generated those from your sodium potassium ATPase pump, and very little on the outside. Okay, these are outside, these are inside. Okay. So once this channel opens, in which direction would your potassium move? Right? These potassiums are going to move out from their high to their low. Okay, so that's a very main important principle to remember. They always go from high to low. Yeah. So they go from high to low, just like water dam opening up, all the water rushing out. Right? So as the potassium channel rushes out, yeah, so all your potassiums are going to be on the outside. Okay, so leaving your inside more negative again because all the positive charges are leaving. So that's why it went from a plus over here, yeah, back to a negative charge inside the cell. Okay, so that's why you can see in your action potential, when we measure, for instance, you'll see a general two phases of your action potential. But let's divide this very carefully. So over here is time on the x-axis. Over here is your measurement of the voltage in millivolts. So your minus 70 here is our resting membrane potential. Okay, membrane potential. Now, when we have our stimulus, okay, so it could be from any type of things, right? So once you have your stimulus, it sends a signal to your neurons, and if this neuron, if this stimulus is strong enough, they will be able to reach what we call the threshold. So here is that second dashed line over here. If the stimulus is strong enough and passes that threshold, here it's gonna go. Okay. So, but if it's not strong enough, it's not gonna start at all. So we call action potential have a all or none principle. For instance, like looking at dominoes. If the signal is strong enough, it will push down one of the dominoes and everything will go. But if it's not strong enough, it's too weak, nothing will start. So this is how our nerve impulses work. So over here, once the strong stimulus passes through the threshold, it starts from minus all the way to a plus sign. So we call this phase the depolarizing phase. Because before here was outside a cell and here is inside a cell. Yeah. So before there was more negative inside, more positive outside. But when your sodium channels open, okay, then it changes, right? So all the positive charge rushed inside, going from a minus 70 to a plus 30. Okay? So this is when your sodium channels open and all your sodium all your sodium ions rushed inside. So reaching this depolarization phase. So they are not polarized anymore, yeah, because they're all rushed in based on your sodium channel. And next you have your polarizing phase, okay, or actually you are called a repolarizing phase. So now we're going to polarize it again because your potassium channels, oops, I'm spelling it wrong, channels are going to open and all your potassiums are going to rush outside to the cell, okay, to where there's low concentration of. So when they all rushed out, then the inside of the cell becomes negative again. So going from a positive back to a negative value. Okay. So there is a hyperpolarization phase right over here where your cells are going to undergo refractory period. So we will see, for instance, two refractory period. Okay. One is what we call absolute refractory period. One is a relative refractory period. It's during the absolute refractory period that you cannot have another action potential going on. So a second action potential will not be able to go on, and this is because your sodium channels are not ready yet, so they are inactive to respond to a different stimulus. But during relative refractory period, you can start, for instance, an action potential if you have a stronger stimulus. Okay. So take a look at this. I know the last part of the video lecture is harder to digest, but you'll be able to do so. Make sure you can link what's happening, for instance, inside the cell, which channels are opening, and how is your ions flowing from their high to their low. Okay, so this will conclude the first part of chapter 12. I'll see you in class for the next one.